Right. Good afternoon, everyone, physical students and online students. So we are about to start biology class today. Last week we were discussing about cells. So we discussed about prokaryotic cells and eukaryotic cells, as well as we discussed about the differences between prokaryotes and eukaryotes. Also, we started discussing about the subcellular components and outer margin of the cell. What is that? That is the plasma membrane structure. Hari. Can you tell me whether we discuss everything about plasma membrane functions also? Hari. So here is one question for homework for this time. Please write this and go. Essay. Structure and functions of structure and functions of plasma membrane. Next week, you have to submit this one. The online students should send me as a clear scan, copy, don't send unclear diagrams, unclear photos. Please send a clear diagram, scan with scan or whatever the scanning app, or else get a clear picture and send it to me. I can scan it. Yeah. Write down the question, everyone. Hi, can I use the board, children? You're done? Write the topic. Structure of mitochondria. I'm going to draw the structure of the mitochondrion. Mitochondria is the plural term. Mitochondrion is the singular term. Okay? Right. So it is a elongated structure. Look at how I draw first. Don't draw anything. Don't draw 3D diagrams. That is not necessary to draw 3D diagrams. In the book, they have given a 3D diagram. So I'm drawing a 2D diagram. Get used to one proper diagram for the exam and everything. You use one diagram. Okay? Alright. So this is the outer membrane of the mitochondrion. Outer membrane is smooth. There are no convoluted nature. Smooth. Yeah, don't draw anything. Don't write anything. Alright. So here the inner membrane is having foldings like this. It is convoluted, right? Alright. The label at the same time. This is outer membrane. Here it is inner membrane. In between these two, this is intermembrane space. This is a double membrane bound organelle. So inner to the inner membrane, there is a fluid filled area. We called it as matrix of mitochondria. Mitochondrial matrix also similar. And inside the matrix, there is a DNA molecule, which is a circular DNA. We call it as mitochondrial DNA. So inside that, there are ribosomes. Which ribosomes are here? 70 small ribosomes are there. Sometimes there are some particles attached to these areas. These are called stalk particles. Correct? Stalk particles are there. And here, these infoldings here, we call it as cristae. Correct? Look at the picture again. Outer membrane is there, that is a smooth membrane. Inner to that, inner membrane is there, it's the convoluted membrane. And in between that, intermembrane space is there. And then inner to the inner membrane, liquid area is there, but that is called mitochondrial matrix. And inside that, there is a circular DNA, which you know that prokaryotes also had the circular DNA, and 70 ribosomes, which are similar to prokaryotes, no? So this gives some evidence for what? This gives some evidence saying that mitochondria was early prokaryotes. They were engulfed by a big cell. Now they are living mutually. Understood now? All right, let's draw this one. Draw nicely with a sharp pencil. Tell Rashida to watch the recording so that will be covered. Done? Right. Shall we write the answer? We are going to write the structure of the mitochondrion. Picture is the must for that. And then we are going to write that. Yes, it's coming. Hi, good afternoon. Okay, please. Right. So, yeah, will you be able to copy this soon before I erase? Right. Others wait a little. We'll finish the one. So we'll write and draw later. I'm not erasing this now. Right. Right. As sentence wise. Number one, mitochondrion is an elongated organelle. Elongated. Within bracket, rod shape. Rod shape. Number two, it is a double membrane bound organelle. Double membrane bound organelle. Number three, outermost membrane is smooth. Outermost membrane is smooth. Next one, but the inner membrane is convoluted due to cristae. Convoluted to form cristae. Let's write like that. Convoluted to form cristae. Convoluted to form cristae. Cristae, the word is here. Next one, cristae increases the surface area. Cristae increases the surface area. Next one, in between the two membranes, the space is called intermembrane space. The space is called intermembrane space. Correct? Next one, enzymes which are necessary for electron transport chain within brackets oxidative phosphorylation. record करा. I contact करा that. Oxidative oxidative phosphorylation are found on the inner membrane of the mitochondria, necessary for electron transport chain are found on the inner membrane of the mitochondria. And this one you will learn in next lesson, so don't worry about this. Since you are studying for the big exam, you will need this. That's why I added this here, right? Okay. Next one. Inner to the inner membrane, mitochondrial matrix is located. Inner to the inner membrane, mitochondrial matrix is located. Inner to the inner membrane, we write like this. Inner to the inner membrane, liquid filled matrix is located. And ඊළඟ එක, inside the mitochondrial matrix, put A, B, C, D and write A, circular DNA molecule, B, 70S ribosomes, C, phosphate granules, C, enzymes necessary for pyruvate oxidation and Krebs cycle, K capital, right? Krebs cycle in cellular aerobic respiration. Hi. So that's all about mitochondria. Can you all read the text given also in the book? Check whether anything is not written. And meanwhile, Sia, draw the picture quickly. Common exam. Oh, then. Exam ever had one. Then, sorry, including 11 for 90. അറിയാമോ ആൻസർ യാത് നേരായാണ് So did we miss anything? Nane? Hi. So we are going to the functions of the mitochondrion. What are the functions given there? Cellular respiration. Mitochondria is specially necessary for cellular aerobic respiration, isn't it? And then what about the other function? Photorespiration. So this is a new word that you will be learning. This is coming up lesson. In some plants, in the photosynthesis, there is some compound form which is a bit toxic to the plant. So that there is a small pathway called photorespiration pathway to make it into a non-toxic product. So in that process, chloroplast, mitochondria, and peroxisomes are involved. So here they are including the functions because we will be learning in future. Okay? Other. Hi, write the function, everyone. Amira Singh. Try the next topic, structure of the chloroplast. Try the topic and listen. Ready? This is having biconvex lens shape. Not an elongated structure, having a biconvex lens shape. That is also a double membrane bound organelle. So that what is this membrane? Outer membrane. So inner to that, there's another membrane that is also smooth. Both the membranes are smooth here. So what is located in between these two membranes? Intermembrane space. Here it is intermembrane space. So inner to that there is a space which is having a liquid fluid. This is called stroma. Stroma of chloroplast. You can just write stroma also, right? Okay. So inside the stroma there is another membrane system, right? So that membrane system we call it as thylakoid membrane. So here are the thylakoid membranes. They stuck together. This one single structure we can say a thylakoid membrane. On these all are thylakoid membranes. On these only photosynthetic pigments and enzymes necessary for photosynthesis are located. Further, so these are found in the matrix. These stacks of thylakoid membranes, we call it as grana. Granum is the singular term. If a particular thylakoid membrane is connecting to grana, we call it as intergranal lamellae. And in the matrix, there is a circular DNA molecule. Stroma also called as matrix, but our lessons we are using stroma word, right? Sometimes in the books you may see as matrix also, right? What are these? 70s ribosomes. Other than that, starch granules also present in the chloroplast stroma, right? Draw this nicely. Shall we write? Right, number 1. This is a double membrane bound organelle. Number 2, it is a biconvex lens-shaped organelle. It is a biconvex lens-shaped organelle. Correct. Next one. Both inner and outer membranes are smooth. Both inner and outer membranes are smooth. Correct. Next one. Inner and outer membranes are separated by inner and outer membranes are separated by inner and outer membranes are separated by a very narrow intermembrane space. A very narrow intermembrane space. One thing we have missed to label here. Thank you, Imana. Circular DNA. Correct? Next one. Inner to the inner membrane, the fluid-filled stroma is located. Inner to the inner membrane, the fluid-filled stroma is located. Inner to the inner membrane, the fluid-filled stroma is located. And inner to the inner membrane, the fluid-filled stroma is located. Next one. Inside the stroma, there is another membrane system called as thylakoid membrane. Membrane system called as thylakoid membrane. Thylakoid membranes. This system consists of flattened and interconnected sacs called thylakoids. Flattened and interconnected sacs called thylakoids. Thylakoid membranes have pigment molecules such as chlorophyll A, chlorophyll B, carotene, and xanthophyll. And enzymes necessary for light dependent reactions of photosynthesis. ඊළඟට thylakoids stack to form granule, granule, granule. Next one. The grana are interconnected by intergranal lamellae. It is the term intergranal lamellae. Next one. Stroma also has we write as a list A, circular DNA, B, 70S ribosomes, C, starch granules, D, enzymes necessary for light independent reaction of photosynthesis. හරිනේ? හරිද? Now read the book also, check whether if we have missed anything. What is the thing? Nothing is missed. Right, okay. Write the functions of the chloroplast. Number one, photosynthesis. Number two, photorespiration. Include that function too, okay? Now tell me, what do you think when you're asked to write the structure of mitochondria or structure of chloroplast? Is it a must to draw the diagram? Yes. So let's say you're going to the exam and this question is given. What would you do first? Draw the structure. It is easy to draw the structure because you can memorize a lot from the structure. You can write a lot from the structure. Looking at this, you can say that there are two membranes, outer membrane and inner membrane is there. Those are smooth. In between those intermembrane space is there. Inner to that, the mitochondria, sorry, chloroplastoma is located in that. Another membrane system is there. Thylakoid membranes are there. They are stacking together to form grana. Grana are interconnected by interconnected intergranal lamellae, and there are 78 ribosomes, circular DNA, starch granules present in the mitochondrial matrix. Many things can be written with the diagram. So that I suggest you to draw the diagram first and then label. Labeling is a must. Let's say you have done the diagram well, you have not labeled. You are not going to get marks, okay? Hurry. Right, we'll give you 5 minutes. Again, go through two structures we wrote before we go into the next structure. We are going to the nucleus next, okay? Where's the paper? Okay. Are you done? Shall we go to the next diagram? Right. So we are going to the structure of the nucleus. So it is like a globe. In that globe, having some pores, right? So we are drawing the 2D structure. So we have to cut the globe. So this is also a double membrane bound structure. The membrane. Intermembrane space. This inner membrane. All together, this is called as nuclear envelope. Correct? So here are nuclear pores. Right. Inner to that, the nuclear lamina is located. In that, protein filaments are randomly found, right? It's not a master draw, so I'm just drawing few protein filaments here, right? Inner to that, nuclear matrix is located. In the nuclear matrix, they are having chromosomes. Chromosomes means DNA plus histone proteins. Chromatin, network of chromatin we can say, right? Network of chromatin. This is DNA plus histone proteins. And here is the structure. What is this structure? Nucleolus. Right? Lot of new things you will be learning now. You have to study well. Harder. Shall we label? Draw and label nicely. Are you done? Let's try. Are you sleeping? Right there, it's a double membrane bound organelle. The double membrane bound organelle. Spherical in shape. Spherical in shape. Next one, about 5 micrometer in diameter. About 5 micrometer in diameter. Within brackets, average diameter. The double membrane cover is called nuclear envelope. Double membrane cover is called nuclear envelope. Cover is called nuclear envelope. Correct. Next one. The inner and outer membranes are separated by separated by a space of 20 to 40 nanometer. 20 to 40 nanometers. Next point, nuclear envelope is perforated by nuclear pores. Which has a pore complex to regulate the entry and exit of substances. Inner to the nuclear envelope, nuclear lamina is located. Next one, it is made up of protein filaments. It is made up of protein filaments which lines the interior side of the nuclear envelope. Next one. Inner to the nuclear lamina. Inner to the nuclear lamina, nuclear matrix is located. Next one. Nuclear matrix is made up of. Nuclear matrix is made up of protein filaments, extended throughout the interior of the nucleus. Next one. Chromatin and the nucleolus are embedded. Chromatin and nucleolus are embedded in the nuclear matrix. Next one. Nucleolus appear as darkly stained granules. Darkly stained granules. Stain like pigments. Darkly colored with dark color. Darkly stained granules with fibers adjoining part of the chromatin. Next one. Chromatin appears as diffused mass in electron micrograph of non-dividing cells. Next one. During nuclear divisions, chromatin condenses and tightly coils and folds. Tightly coils, folds, don't write folds, but tightly coils and form. Yeah. During nuclear divisions, chromatin condenses and tightly coils and form threads called chromosomes. Threads called chromosomes. read this and read the given text also in the book. nuclear structure. Had- Do we have to discuss the paper also? I thought of starting at 5:30, 5:30 to 6:30. Time is enough, ne? I will do one more and go. Honey, try the topic. So now double membrane organelles are over. We are going to single membrane bound organelles or structures. Haridan. So I'm drawing something attached to this nucleus. There is a small organelle which is attached to this. What is this structure? This is having ribosomes attached to it. This is rough ER. Correct? This is smooth ER. I didn't ask to draw, paste the pictures. Don't forget to paste the pictures that I have given to you later at home, right? Mitochondria, chloroplast, and nucleus given. And this we are going to learn about endoplasmic reticulum. Write the topic, everyone. Endoplasmic reticulum. There is a particular reason that we are writing and drawing these structures. These are always question papers, right? It is connected to the nuclear envelope. So here is a nuclear envelope, outer layer. What are these structures? What are these structures? Ribosomes. Functions. Didn't we discuss functions of nucleus? Please add the functions of nucleus also. We'll quickly go through those also. I forgot to give that, ne? Before giving this one. Uh, Functions given there, control all cellular activities. Next one, synthesize DNA to produce new nuclei for cell divisions. Next one, synthesize rRNAs and ribosomal subunits required for protein synthesis to nucleolus. Next one, synthesize mRNA and tRNA according to the information present in the DNA. Store and transport genetic material. Study now itself those also. Draw this structure, and here are the transport vesicles. These are budding off from this ER, from smooth ER and rough ER. The edges budded off as vesicles, right? Those are called transport vesicles. And here the lumen, the cavity is the lumen, right? The proteins which are synthesized by these ribosomes are secreted to the lumen, okay? Then they are incorporated into small vesicles and transported towards the Golgi. Finish. Write about smooth ER and rough ER. Write the topic rough ER. It is a single membrane bound organelle. Samai, don't forget to paste the pictures, okay? If you cannot recognize any, just ask from me. Number 2, it is continuous with the outer membrane of the nucleus. It is continuous with the outer membrane of the nucleus. Next one. It forms a network of internal membranes. It forms a network of internal membranes and forms flattened or tubular sacs. And forms flattened or tubular sacs. Flattened or tubular sacs. Next point, these are called cisternae. Cisternae, the word is there, the picture. Next one, interior of these sacs are called lumen of the ER. Next one. There are two types of ER. A, rough ER. A, rough ER. B, smooth ER. Next one. Mm, in the RER, numerous membrane-bound ribosomes are present. Numerous membrane-bound ribosomes are present. Next one. SCR is a network of tubular sacs without ribosomes. SCR produces transport vesicles. Hurry. Let's go to the functions of rough ER and smooth ER. Everyone come to the functions given in the book. Rough ER, transport proteins synthesized by bound ribosomes. That means the proteins are synthesized and secreted to the lumen. They are transported within, right? Next one, synthesizes glycoproteins. What are glycoproteins? Carbohydrates bound to proteins. Next one, produce transport vesicles. It's a common function for both, right? Both rough and smooth ER. Synthesize membrane phospholipids. Facilitate the growth of own membrane by adding phospholipids, proteins, and carbohydrates. Therefore, it is called membrane factory. The factory is producing whatever the necessary things now, so that produce the things in the plasma membrane. This is involved, right? Next one. The functions of smooth endoplasmic reticulum synthesizes lipids including oils, steroids, and phospholipids. Metabolism of carbohydrates, produce transport vesicles to transport within the cell, involves in detoxification, stores calcium 2 plus ions. Okay? All right. Study now itself and copy the functions in both smooth and rough ER. Imana, what did you miss, darling? I don't have the numbers in my sentences. Can you just give a rough idea so that I can read? Where did you miss? First one. He is a single membrane bound organelle. The numerous one, I said that in the RER, numerous membrane-bound ribosomes are present. In the RER, numerous membrane-bound ribosomes are present. Good. Right, let's go to the paper discussion then. So you received the marks now. So the online students, only Amarsha's paper I have marked. We'll send you that few Amarsha, and Ilki's paper I think I didn't mark. I had to check it properly. Imana, you were not there for the paper, no? Hari. So I'm going to the paper. So do you have the soft copy with you? Shall I repost it to the group so that you can get it now? Wait. Yeah, the papers are not usually shared. Please do not share with anyone else. So this time only I'll be sharing the PDF with you. Hari, look at the paper now. The first question is asking what are the characteristics of living organisms? First question, actually, that I have taken any of 6 out of the 7 characteristics. Half into 6, 3 marks. Okay. Order and organization, metabolism, irritability and coordination, heredity and evolution, reproduction, growth and development, adaptation. What are the 4 main biomolecules found on Earth? Carbohydrates, proteins, lipids, nucleic acids. Half into 4, 2 marks. Write the organizational levels of living matter in the correct sequential order. When I teach this also, I told that this is a confusing place. I asked from few people, they told to start with molecules, but this time also for this I took from cells. I will recheck about the idea, right? What they have given, because in the 8th unit environmental biology lesson, they are starting from the cell, okay? Yes. So cell, tissues, organs, organ system, organism, individual, population, community, ecosystem, biospace, the order. Correct order should be there. Correct number of categories should be there to obtain 1 mark there. So this kind of questions in the middle, if you have written wrong things, the total mark is lost. What is the most abundant group of organic compound on earth? Carbohydrates. What is the inorganic compound? Water. Take the monosaccharide with least number of carbon atoms, glyceraldehyde. Okay. Write down the answers if you not have written the correct answer there. Draw the structure of an amino acid. So I'm going to draw it and show you. C R C double bond O OH. You can write it in the other way also. NH2 and H. Arne? You have to label also what glycine is asked. So, sorry, this one hydrogen should be there instead of R. No need to label, just you can leave it as this. Right? Instead of this, you may write COOH. Instead of this, you may write NH2. Right? Draw and show how a peptide bond is formed between 2 amino acids. I'm going to draw it and show you. Previous one, 2 marks for the structure of glycine. That's the simplest amino acid. Let me. Peptide bond should be labeled there. Sleepy, not today. I know. This lesson is not that interesting. Hari, okay. Next one. That for that question, marks are allocated like this. 2 marks, okay? Next one. Name the major biological polymers and state their major elemental composition. Biological. Type it. You have to write the monosaccharide example. Type, if they ask type, that is triose. Not that wrong also. That is correct, but still nitrogen is connected to the bond. Not wrong actually, can give mark if you want. Mark it with the pencil. Hurry. For you can't give, huh? The monosaccharide mask. The name. Hurry. The biological polymers, there are 3. Carbohydrates, if you write carbohydrates, you are not going to get marks. Why? Why is that? Carbohydrates, you must write polysaccharides. Just carbohydrates is not going to get marks. The element composition is CHO. Proteins, CHONS. Nucleic acid, CHONP. Had the 3 marks, half into 6, 3 marks. Define the following levels of protein structure and give one example for each. Secondary structure, the correct definition should be there, halfway written, not going to get marks. Secondary structure, the primary structure of a single polypeptide chain coils and folds as a result of intramolecular hydrogen bonds between the oxygen atoms and hydrogen atoms attached to the nitrogen atoms. So 2 marks are given there. One part of the definition, 1 mark. Second part about intramolecular hydrogen bonds, 1 mark. Next one, the secondary polypeptide chain bends and folds extensively, forming a precise, compact, unique, functional and 3-dimensional shape, 2 marks. Quaternary structure, aggregation of 2 or more polypeptide chains involved in the formation of one functional protein, 2 marks. Quaternary structure, that one I told. Mention the types of monomer molecule of the following organic compound: cellulose, glucose, pectin, galacturonic acid, inulin, fructose, glycogen, glucose. Fourth one, out of the above compounds A to D, mention 2 types of structural and storage compounds. Storage compounds, cellulose and pectin. Storage, inulin and glycogen, 2 marks each. Write an experiment to identify the major organic compounds present in the egg. The test is biuret test. You have to identify the test first. Next one, a solution of egg is prepared, 1 mark. 2 cubic centimeters of solution is added to an equal volume of 5% KOH solution. 2 cubic centimeter, 1 mark. Equal volume of 5% KOH, 1 mark. Few drops of 1% copper sulfate, 1 mark, is added to the solution and mixed, 1 mark. Color change is observed, 1 mark. Color change is observed, there's no mark allocated for that. Do you want me to read it slowly? Write down there. A solution of the egg is prepared. Or as you may say that, egg solution is prepared. Number 2, 2 cubic centimeters of the solution is added. 2 cubic centimeters of the solution is added. Next point, to an equal volume of 5%. To an equal volume of 5% KOH. 5% KOH solution. And mixed. Next point, few drops of 1% copper sulfate, few drops of 1% copper sulfate is added to the solution and mixed. Next, the color change is observed. Hi. Answer the following question related to given compounds below. State the monosaccharide of the above compounds, glyceraldehyde, ribulose, 2 marks. What compound is the intermediate product of photosynthesis? Glyceraldehyde. Which compound is found only in animals? Lactose, testosterone. Number 4. Which one is the steroid hormone? Testosterone. Which compound is the storage carbohydrate? Inulin. So 1 mark in the sense there are 40 points in this question. Those 40 points you have to multiply it by 2.5 to get mark out of 100. That is how the marks are given in the exam. Usually the half marks are not given. I wanted to give more questions so that I have added more points there. Okay. Usually 1 mark for each marking tick. Okay. Second question. The following picture shows a part of a DNA molecule. Write the answers using the given diagram. Fill the following table by the sequence of nitrogenous bases of the lower DNA strand. So the given strand is GAA-TTC. So complementary DNA strand is asked CTTAAG. All right, 1 mark for that. Next one, there is a diagram of the DNA molecule is given here. You are asked to label the parts. First one, second one, third one, fourth one, and fifth one. First one, thiamine nitrogenous base. Thiamine only. Is there anyone who has written thiamine only? It also can be considered. Cytosine nitrogenous base number 2. Number 3, deoxyribose sugar. You can't just write sugar or pentose sugar. Can't write like that. Next one, phosphate group. Fifth one, deoxyribonucleotide. Just nucleotide won't get marks. Deoxyribonucleotide. The third one. Let me check. Can write only one, but only half mark is awarded, right? Hurry. Next one. How many nitrogenous bases? Actually, it should be nitrogenous base pairs are present in one turn of DNA molecule? 10. Who introduced double helix structure? Watson and Crick. What is the structural unit of DNA? Deoxyribonucleotides. 1 mark for each. For the labeling of the diagram also 1 mark for each label. Next one. What are saturated fats? Fats which do not have any double bonds in their hydrocarbon chain. Fats do not have any double bond in their hydrocarbon chain. Okay? Or else only single bonds also you can mention. Example, butter. What are unsaturated fats? Give one example. Fats which have one or more double bonds in the hydrocarbon chain. Example, coconut oil. Okay, no? Hi. One mark for that also. Next slide. Why amino acids are considered as amphoteric? Amino acids are having both acidic group, which is COOH, and basic NH2 groups, which within the same molecule. One mark. Next one. What are three major components of a nucleotide? Nitrogenous base, pentose sugar, and phosphate group. Fifth one, write three functions of nucleotides: acting as electron carrier, energy carrier, acting as a coenzyme, acting as a reducing agent, as oxidizing agent. Electron carrier also can be added. 1.5 marks, 0.5 each. Take two common features of monosaccharides: sweet in taste, water soluble. Second one, what is the reaction type occur during formation of disaccharide by joining two monosaccharides? Condensation reaction. Which property of phospholipid molecules causes the organization of them as a bilayer in the plasma membrane? Amphipathic nature. Name a lipid found in plasma membrane of animal cells only. Cholesterol. State major function of each following protein in living body: keratin, forming skin, hair, nails; serum albumin, transport fatty acids. There are 20 points in that question. 20 into 2.5, 50 marks are allocated for that question. Got it? Third question. Write three differences between RNA and DNA. It may be structural or chemical differences you may write. Adipute, is there any problem? No? Three differences. DNA, the structural unit is deoxyribonucleotide, RNA, the ribonucleotide. DNA thymine is present, RNA thymine is absent. DNA uracil is absent, RNA uracil is present. And other than that, you may write single strand RNA, double stranded DNA. Any considerable answer can get points. Next one. Write four main properties of water that maintains life on Earth: cohesive behavior, ability to moderate temperature, expansion upon freezing, versatility as a solvent. 4 marks. Previous one, 3 marks. Name the elements which account for 96.3% of the body mass in humans, OCHN. Write functions of DNA in living matter: store and transmit genetic information from one generation to next generation. Store genetic information for protein synthesis. 2 marks. Mark, ne? You have read it? Sorry, being 2.5. Here, that means 68. Right? Tell me at the end to change the final. Right, next one. Your mark also, can I change that previous one? Wait. Which one, darling? This is correct. This one, how many marks I have? This is the end of this question. No? 2 means 5. 78. No? At the end, we'll change 5. No? Yeah. 1 mark. No? 1 mark means. Sorry. 1 mark means 2.5 to 70. No? Not writing answers. Next question. Write one functions of each type of RNA, rRNA formation of the ribosome, or else provide the site where polypeptide chains are assembled. tRNA, transportation of amino acids to the site of protein synthesis. mRNA, copies genetic information stored in DNA molecule as a sequence of nitrogenous bases. 2 marks for that, 3 answers. Okay. Next one. Given below is the structure of a biological polymer found in living matter. What is the type of biomolecule represented by the given structure? Protein. Label A, B, and C. A is peptide bond. B is amino group or N terminus. C is carboxyl group or C terminus. Write the element composition of the above molecule, CHONS. What is the structural unit of the above molecule? Amino acid. Why this molecule is considered as a biological polymer? Because it is made up of large number of structural units called amino acids. Amino acid part is not mandatory to mention. 2 marks for each, last two, 5, 4, and 3. And for the second question, 3 marks. First question, 2 marks. Okay? B. I'm telling about B. Had. Next one. Write 3 functions of polysaccharides. Storage compounds in organisms, form structures in organisms, acting as an energy source. Any correct answer can get marks. Do you have any other answers which is not marked? Next one. What are structural polysaccharides? Polysaccharides which are forming structures are called structural polysaccharides. Next one. What is the energy carrier molecule in living systems? ATP. Name 2 organisms which store glycogen, humans and fungi. Yeah, sure. C. 2 means 5 marks. Sorry. At the end, we'll change all. Right. Okay, now next one. Write 3 functions of lipids. Food reserves as energy source. Maintain the fluidity of the plasma membrane. Act as signaling molecules. Found as components of animal cell membrane. 3 marks. Any 3? 3 marks. To the. Another 1 mark is added. Plus 2.5. Okay. Next one. Name 2 lipid hormones, testosterone, estrogen. Any other answers you can write? What are the other lipid hormones that we discussed? Estradiol, testosterone, aldosterone, cortisol. Okay. 1 mark the fourth one. Yeah. 1 mark here. So 0.5 is added. Ne? Yeah. 0.5 into 0.5. 0.5 into 2.5. I will add 1. Ne? Are add this one. Cortisol is correct. Function of making the structure of cell membrane. Okay. Yeah. Correct. All right. Next one. Name 2 protein hormones: insulin, glucagon. Any other protein hormone can be written as the answers. Next one. What is adaptation? Peculiarity of structure, physiology, behavior that promotes the likelihood of an organism's survival and reproduction in a particular environment. 1 mark. What is irritability? Ability to respond to stimuli from both internal and external environment. 1 minute, we'll read this and come. Briefly explain how cohesion forces are important for living organisms. Cohesion forces are forces between water molecules. This helps to transport water and minerals in the xylem. Cohesion forces also give high surface tension to water. Animals such as water skaters can walk on water. Next one. Before part B, what is your problem? You are not written the complete answer, no? Peculiarity of structure or physiology. Physiological behavior name physiology. Right? Okay. You have write as it is. Put it. Next one. Physiological behavior. Only that place you have altered. All others correct. For today we'll give. Hurry. That lesson. Yeah. Then 2 marks. Ne? 1. Ne? 2.5. Alright. Next one. B, part B. You have to draw the structure of a nucleotide there. So you may draw it like this. Alright? Better if you can label it. This is the nitrogenous base. This is the pentose sugar. This is phosphate group. Okay? Alright? Next one. A scientist analyzed a section of DNA from sea urchin. Section was 249 base pairs long and 17.5% of the bases were cytosine. Find the percentage of thymine bases present in this section of DNA. Show your working. So you have to do it. How many of you got this correct? Good. What happened to you? Wrong. Correct. You? Okay, we'll do. Half is correct. Whose half is correct? Right. So they have told that 249 bases are there. Base pairs long. Pairs are there. It says that cytosine is available 17.5. Cytosine amount should be equal to guanine amount. So it is also 17.5%. So if we add this together, you will get what is it? 35% C plus T. So the rest is T plus A. So 100 minus 35, what is it? 65% is guanine, so thymine plus adenine. So that 65 divided by 2 will give you number of 32%, 5. This is the thymine percentage. Okay, got it? Very easy, no? All right. Write this down if you have not written correctly. Next one. Again, write the complementary RNA sequence for the given DNA section. So we are going to write the RNA sequence. U, G, G, C, thymine is not there, U, so G, E, E. RNA. Next one, starch is a carbohydrate that breaks down into glucose. What is the type of reaction that breaks down starch into glucose? Hydrolysis reaction. What are the major biological organic polymers which contain nitrogen? For proteins and nucleic acids. What is an anabolic reaction? Given example, making large complex molecules using smaller simple compounds. Example, photosynthesis, 2 marks for that. What is a catabolic reaction? Given example, breaking down of larger complex molecules to form smaller simple compounds. Example, respiration. Why lipids are not considered as macromolecules? Because they do not have large molecular weight. 1 mark for that, previous to 2 marks, 20 points into 2.5, 50 marks for that. Previous question number 3, 100 marks, this one 50 marks. So you are getting marks out of 200 for the structured essay paper. 4 questions are there, 200 marks are there. Now check the total out of 200 and tell me if there are changes. You have to calculate and tell me. මෙතන පුතේ 0.5 නේ ඇඩ් වෙන්නේ 1 නෙවෙයි මෙතන 0.5 ඇඩ් වෙලා තිබ්බ already 1 තමයි ඒ කියන්නේ තව 0.5 තමයි ඇඩ් වෙන එකම 1 කියලා දැම්මේ 1.25 දාන්න බෑනේ ඒ නිසා හරි බලන්න ටෝටල් එක අරන් කිය වෙනස් වෙනවා කිය Hmm. That means 1 mark I have added here. No? Yeah. In a definition, one word is yeah, but the structure they say they are marking very strictly. Okay. Some certain small points we can consider, but like let's say the definition of adaptation or whatever. Hmm. Hmm. 20 into 2.5. 50 no, but 2 into 2, 25, 50 no. How come 60? Mathematical errors. 2.5 into 20, 50. Yes. Should send to mathematics class. That's fine. Final, you have added this last one also. So how many marks are added now? 6 marks means not the total. So here six marks out of this, out of 206, na? Mm. That is given out of 200. This 2 given out of 300. Okay? So that means all out of 500. Right? Only 4 structured essays are there, Anupute. First question 100. Right? Second question 50. Third question 50. No, so third question 100. Fourth question 50. It is 300. We should send to mathematics class of Sudhir sir now. It's 200, right? Not 300. 200 then 250. 300. So my mathematics is wrong now. 300, 300. So I divide by 6. So get the total and divide by 6 and check whether this is changing or not. Sometimes it may not change. Yeah. Take all the total and divide it by 6. With the essays I'm telling, you have to practice a lot. Add all and divide by 6. In that you have to count and 6. I don't remember one by one. 5 marks added. What was your total marks added? No, no, no. Structure they say? No, no. Total marks added now. 6 marks. These 6 marks. No chance of adding 5 marks. Oh, one. Only 1 mark. So that's all. Enough. Alright. Okay. Additions? No changes. Any additions? No. How do you feel about the paper? Is it good? Happy about marks? Easy mistakes, no? 66. Wait. Go home and redo the corrections, right? So that you can learn a lot. At least if you repeat these papers 2-3 times, you can go for a good mark in your first exams in the schools. Have you had any exams at school? Not yet, no? All right. We have time, we'll go for another organelle also. Write the topic, Golgi apparatus. All right, so we are going to draw Golgi apparatus there. This is a single membrane bound organelle. There are vesicles coming from the ER. What is that vesicle? We labeled previously? Transport vesicles. However, since this is getting transport vesicles from the ER, this side we call it as this side. It's wrongly labeled in the book. I don't know whether they have changed it or not. The new version, not changed it. So it's wrong. When you read the text, you will understand this side is cis, okay? And the other side, there are vesicles produced from this end also. This vesicles, we call it as secretory vesicles. So These sacs-like structures, again, we call them as histone. And the lumen is here inside. This side, which is producing secretory vesicles, we called it as trans side. Let's draw. Right. Go and read the structure of Golgi apparatus and see what I told is correct or not. Cis trans structures, cis trans sites. Correct. Then we'll write the answer.